Hi, um, this is the third video of the EcoEvolutionary talks. Um, I'm Cíntia Freitas, postdoc fellow at the Universidade Federal do Paraná at the Print Caps program. Um, and today I'm going to present some insights on the phylogenetic structure of the palm community, still in Reserva Duque, Manaus. Okay, and again, if you have any doubts, please email me um, or leave comments down below. So today I'm going to talk about this paper that we published in 2014, and in here we investigate the phylogenetic structure of palm communities in this region of the central Amazon right here, Duque Reserve. You guys know from the last videos I posted here. So community ecology, even today, even after let's see 6 years, is still lacking a set of central theories that synthesize its general principles. So by the time I was writing this paper, the phylogenetic structure of palm community in the central Amazon changed along a hydroedific gradient, and here you can see the full reference. The phylogenetic structure of communities was a big hope to understand the sorting of species at our local communities. So here we can see again the Pepe grid, but now instead of 30 plots, I gathered data on the other 42 plots. So in this particular study, I used 72 plots, sampling palms in these plots. So just quick going here. For that specific study, I needed to produce this phylogeny of the local palm community. It's actually not a phylogeny; it's a topology. Since it's not dated, it's not calibrated with any calibration points. So the molecular systematics, they get DNA and sequence the samples, the species, now the samples, in order to get a molecular phylogeny. However, a simple topology based on published data is what we ecologists sometimes use, or use back then, because things are going so fast that ecologists now are using molecular phylogenies to test some cool hypotheses. But back then we used to have only those topologies. So that's the type of data we used to have to do some inference in the local ecology, including evolution on our studies. So, and we didn't have DNA, of course, information for many of our samples here represented by those black dots. So we have information, not exactly DNA, but we have like published phylogeny. So all the white dots right here are like actual relationships, phylogenetic relationship based on molecular studies, published molecular studies, and the black dots are the information, mostly because never were those species were never collected in Amazon. Even if the phylogeny of bacteria was published by the time, it did not include any of those Amazonian species. So the black ones are the ones that we inserted artificially at the nodes that we knew were the closer related. So we do this in R, and it's not the best solution, but was a solution we had back then. so it was acceptable. And to assess the uncertainty in all the calculations I'm going to show you guys next slide, we randomized the tips within each genus, and we got 100 trees and calculated all the parameters using those 100 trees just to assess the uncertainty. So here we have Mauritia, Mauricella, and Idiatella, Socratea, which are like the most basal, and we have Geonoma, Eospati, Euterpis, Rhinocarpus, and then we have here Ataleini, Cocosi, with Siagrus, Atalia, Desmoncus, and here we have Bactridini subtribe. So our questions. Sorry, before that, we calculated the relationship, the closeness between each pair of species. So we calculate the net relatedness index, N or I. So we calculated those metrics using the this is an overall relatedness, it's the index of how close related those species are. And okay, let's just see here. In this case, we calculate using the equations, this one and this one here, the nearest taxon index, using the length of the branches, like this. So we measure the number of substitutions here, and this is the length of the branch, and we calculate this mean. So the net related index captures the deepest structure of the tree, while this one, the NTI, calculates the structure of the tips in the phylogeny. So it is the simple mean of the distance in the value of the branch lengths between a pair of species, in the case of MPD, and the mean in the values of the branch lengths of the nearest taxon in the tree, in the case of MNTD. So those means are calculated in each plot and in the whole grid you saw in the last slide. So positive scores indicate phylogenetic clustering, or the species, the close relatives who occur more than expected by chance, and negative scores in both cases indicate phylogenetic overdispersion, where close relatives occur less than expected. And our questions. So it was a simple question. So do close relatives co-occur more or less than expected by chance, and by that we calculated that indexes and this local scale. So if they co-occur more than expected by chance, the prediction is that the environment filters some species with similar traits. So the environment selecting species with similar traits. If they occur less than expected by chance, competitive interactions between species with similar traits eliminate the poor competitor. So here see two important premises. First, species are close related. Okay, well, close related, and we are assuming that they have similar traits just because they are close related. So this is called phylogenetic niche conservatism. But it's not always the case. So in the first case, and under niche conservatism, phylogenetic clustering is commonly thought to be the result of environmental filtering favoring close relatives that share the same adaptations, while overdispersion is seen as a result of limiting similarity among closely related species. So they are similar but it's slightly different. So this limiting similarity is a result of them competing for the same resources but not excluding each other. So patterns in phylogenetic community structure um, would help ecologists to separate the different niche processes that may promote species coexistence, right? So this species coexist because they are suffering the same environmental pressure and they have similar traits, so they filter only those occurring here, and the competitive interactions eliminate the poor competitor, or in the case they change a little bit so they can coexist. Okay, let's just see this up close. So such as those two concepts of local ecology, the competitive exclusion and the theory of limiting similarity, both explaining species coexistence at the local plot. In the competitive exclusion hypothesis, two species with similar traits will never occur, and the best competitor will exclude the lower competitor. So here, either way, we have the same species with us in the environment that's homogeneous. One individual will exclude each other, and within species, we have here, if this species is occurring together with this one that say it's a better competitor, it will be excluded because there is absolutely no way you can share this homogeneous environment, because this one is a better competitor. So for those species to co-occur, and then the richness, diversity of the local plot to increase, there will be slightly different association here, and this is the same principle. This is slightly different environmental gradient, so the species can use different parts of the niche. The same principle is the limiting similarity. So species are slightly different and occupy slightly different parts of the gradient, so you can co-occur and you don't see exclusion right here. So the two species would change a little their traits, or they just occupy different parts of the environment to avoid competition, and then they would occupy slightly different portions of the gradient. And in this case and in this case, exclusion will never happen and diversity will increase, right? So coming back to my questions, do close relatives occur more or less than expected by chance at local scale? So we have here the indexes. Remember, this one it's like seeing the deeper relationship between species in the phylogeny, and this is more related to a pair of species in the tips of the phylogeny. And we have abundance and presence and absence, just to see if this changes any pattern. So I'm going to talk about only for presence and absence. We found some significant relationships. So the gray dots represent plots where the species didn't occur more or less than expected by chance. The pattern was not different from the random. So actually the species are a random subsample of the regional pool of species. But right here we have positive scores indicating clustering. So in those plots specifically, species are more co-occur more than expected by chance. And right there we have some over-dispersion, where species co-occur closer relatives co-occur less than expected by chance. Okay, and we are going to analyze those plots up close in a minute. Regarding the second question, do species possess traits that are conserved at the community scale? So I sampled a lot of traits in the literature, and a trait that is conserved, they are present. The trait is present in a clade, in a monophyletic clade, and we call it conserved in the phylogeny. The only trait that we found to be conserved was the presence of spines, and it's funny because the spines are all present in the Bactrididae subtribe, excluding all the others, which was kind of obvious for one that knows palm taxonomy. So we calculate the K statistics of Blomberg. Values of K greater than 0 and smaller than 1, which is not the case here, indicate trait lability. So the traits, close relative present traits, that are different, that are more different than expected by chance, while K values greater than 1, which is the case here, indicate trait conservatism. So close relatives present traits that are more similar to each other than expected by chance, which is the case of spines here in Bactrididae. So my next question is that if the PCS, or phylogenetic community structure, in each plot change along the most important environmental gradient. So this is just to show how we can merge environmental gradients ordered by the value, species occurrences. Here's the name of species in the school graph. It's called Poncho function by Dambros, by Christian Dambros. And we have here the phylogenies, and we can infer some cool stuff from this graphic. So here in the graphics we see that the clay content gradient and the honey gradient, that's the height above the nearest drainage, it's a surrogate for distance to the water availability. We see that as the first graph showed, and this open circles right here, the phylogenetic community structure doesn't differ from the random, so this is all random, right? And this is values that are significant lower than 0, and here also significant clustered and over dispersed. So we see here also significantly clustering and significant over dispersion. Here the black circles are significant values, showing that mostly in the bottomlands here, lower hand values and lower clay content values in the sandy bottomlands. NRI and NTI were related to those variables, and compared to upland plots right here, those bottomlands with sandier soils sometimes contain species that were phylogenetically less related than expected by chance, and here with negative index values. So these overdispersions disappeared for hand values over than 10 meters and soil clay content above 10%. And this is very congruent with some studies in the same area, that there is a lot of variation in the bottomland and the species composition, and towards the upland this variation disappears, so it's more predictable, the pattern. So, and see, we see that bottomlands, which are, I say, not suitable for all the species, so there's a lot of specialized species living down there. So here we saw a random phylogenetic community structure overall in Duke Reserve. A lack of phylogenetic niche conservatism in the sense that only one trait was conserved, and evidence for slightly little effect of environmental gradients sorting species at the bottomlands. I would like to thank you all the institutions that made this ecoevolutionary talks possible, and thank you so much. I hope you like it, and see you next time.